Well, thank you for joining the very last edition of this academic year's Westwood Lecture.
Thank you.
And this is the eighth one. This AY, outstanding speakers from a broad range of intellectual pursuit. And today, with Majid Kazemian, we have representation actually from three different parts of campus. In the College of Ag, Department of Biochemistry, College of Science and Engineering too, Computer Science Department, and also in the Cancer Center too. About drug discovery from I remember from In Silico to In Vivo, from computer to clinic. This is, I know, a stressful moment for many researchers, thinking about our research funding, in particular direct and indirect. We also understand this is a moment of transformative opportunities to create new mechanisms, architect new pathways, and build new muscles in generating public-private partnership in funding and supporting and translating the impact of fundamental research. We're very proud of our life science colleagues, proud of the collaboration, in this case, between colleges and departments. Proud of our physical AI, proud of our cancer center, and proud of our collective effort by so many faculty, staff, and students. Keeping focus even when the moments might be stressful. And we are here to provide a calm and supportive environment, helping you to create new pathways, and keeping our outstanding researchers to be as successful as possible. That's a very long-winded introduction, but I know that there are a lot of things on your mind, and we share the thoughts, but we also would like to create new opportunities together. I'm looking forward to today's Westwood Lecture. Majid, please.
Thank you so much. Thank you for that kind introduction, and thank you everyone for taking the time to come to this lecture. It's very honored to give this lecture. So our lab focuses on answering two key questions. And the first question is, why do people often react differently to the same disease? Even though we control for age, we control for the sex, we control for stage of the disease, still they respond differently. For example, here, the blue line shows patients which have bladder cancer, and their 15-year survival, if I could get rid of this right here, their 15-year survival is about 75%. Another group of matched patients, their 15-year survival is only 25%. So what determines these differences? And if we could understand this, our second question is, can we intervene? Can we, for example, identify the drugs that could push this poor outcome or poorer survival to better survival profile? And I would like to argue today that computational methods would allow and would help us to answer both of these questions. But this is thanks to generous donation and selfless donations of thousands of patients donated by a specimen, and hundreds of billions of dollars of taxpayer money going to generate data, including databases, medical records, genetic data from these patients, having the drug information, having the published literature that you all contributed to. And in the case of cancer, having big databases where we have genetic information, we have tumor microenvironment, and so on. So with this in mind, let's answer the first question. Why do people often react differently to the same disease? And I would like to suggest that this is because the difference of the cells between the survivors and those with limited survival potential. And cells are like these old radios, and imagine the genes to be these knobs. And some of them are dialed up, some of them are dialed down. This is basically our cells for our broad audience, with the exception that we have about 30,000 of these knobs in each of our cells. So there are a lot of these things that could be tuned. And maybe those that are survivors have a little bit different tunings of these knobs. Maybe they have, for example, a little bit up-tuned of gene 1 or knob 1. They have a little bit down-tuned of the knob 2, and so on and so forth. So if we could identify how these knobs are fine-tuned, potentially we could identify drugs that could modulate these a little bit and fine-tune them, therefore enhancing the survival the way that we want. So that's exactly what we did. So with this in mind, we now looked at all the cancers that are available and asked, like, if we look at the good survivors and those individuals that have limited survival, what are those dials or those knobs that are different between these two groups? And what we saw is that there are hundreds of those knobs that are dialed up in the good survivors, and hundreds of those that are dialed down a little bit in those good survivors. For example, we found out that if we look at one of those specific knobs or genes, that in this case called TCF1, if it's on, you would not get those polyps in the colon that causes the colon cancer or colorectal cancer. If this particular knob is all the way down, then you would have a chance of getting a lot of these polyps that could start enhancing your risk of getting the colorectal cancer. So, all right, we now have this knowledge by comparing these two groups of patients and all of those genes or knobs that I mentioned. Can we intervene? And to answer this, we go back and look at the knowledge that we have. For thousands of the drugs, we already know what genes they dial up and what genes they dial down. For example, this pink drug, we would know that the genes that are up are specific genes. And maybe for this orange drug, we know there's a different profiles and different tunings of these knobs. So having that information, now the job is very easy. So the idea is that, can we take the drugs where we know it dials up or down these knobs and match with the things that we want to survive better? So all we'll do is, like, can we, for example, dial up the gene 1 a little bit? I don't think that this is going to go up, but we'll dial up the gene 1 a little bit for the survival, we dial down the gene 2, and so on. So can we fine-tune these with the drugs that we have? We have one drug, for example, that does this, another drug that does something else, and we'll search through all of these drugs and order them on how could they change these tune-up. So now, the things that could have taken years in the experimental setup or in the labs, within a couple of hours, you'll have this results where for each cancer type shown in the x-axis, bladder cancer, breast cancer, etc., you have list of all the drugs that potentially could enhance the survival in that particular cancer type. On the top of these lists are some of the things that are already known or are in the clinical trial for these particular cancers. So what caught our attention here was a new drug that was never used in the context of bladder or cervical cancer. and that's called CDK9 inhibitor. So we found that that is the drug that potentially enhanced the survivor or enhanced the survival for these particular patients. So that's great because other CDKs have been implicated in disease, but CDK9 has never been implicated, specifically because the gene itself is never upregulated in these cancer types. So we said, well, let's test this. Let's see whether it works. So we took some of the patient samples that went under bladder resection or bladder surgery to remove their bladder and the tumors with it, and we treated this with the CDK9 inhibitor. And what we saw that with an increasing dose of this drug, the tumor growth is reduced. So this is particularly repeated in multiple patients and multiple cells from the patients. While no other CDK inhibitor, CDK4 or 6 or 2 inhibitor did this. So that's great. That's the first step for us to go through this process. So what's next? Does this work inside the body? Because there are many of the things that work in the dish inside your incubator, but this may not ever work inside the body. To test that, we turn back to a mouse model where we place some aggressive form of bladder cancer under the skin of the mice and watch the tumor grow. So in this case, you can see that within the 5 days from day 15, where the tumors appear, up to the day 20, the tumor aggressively grown in size in the blue line. Each of these lines are the tumors from one individual mice that's shown in the blue. If we now treat with this particular CDK9 inhibitor, you can see that the tumors no longer grow. Does this specific to the drug? Well, one way to answer is that if we stop the treatment, all of those tumors start back and growing back up. So wonderful. So in this case, we have a very specific drug that could potentially restrict the growth of the tumors. Wonderful. So what we know is that dogs also have aggressive bladder cancer. And they naturally get these bladder cancer due to many factors, including genetic factors, but also including environmental factors like herbicide and pesticides that are applied to the lawn cares. So the question was, are the things that we see in human bladder cancer using the CDK9 inhibitor, does it also apply to the dog with aggressive bladder cancer? And of course, dogs are our beloved family members. We can't just get a drug and test on them. That's not the way it works. So we need to first show that these are safe drugs, maybe inside the lab dogs, but whether they work on the cancer cells from the dogs, and so on. So we were lucky to have a collaborator who's sitting here, who gave us one of these cancer cell lines from specifically dogs that have aggressive bladder cancer. And we treated them with CDK9 inhibitor, and we saw like exact same response as the human aggressive bladder cancer. So that suggests that probably these two cancers are similar in terms of responding to this particular drug. So the next step was, are these drugs safe? Of course, we needed to show in the mice, and we had already shown in the mice the dose that we had established. But we had to also show in the lab dogs, before going to the pets that get aggressive bladder cancer, that this drug is safe. So in this case, I called up this company who took this particular drug to a safety clinical trial in human and asked them whether they have the drug available and whether they could share some of the drug with us. And they said like, oh, we have already done the lab dog safety trial. So we have all the safety stuff in the lab dogs. And we are happy to share with you the drugs. And then they actually donated hundreds of thousands of dollars worth of the drug for our research to run this. So now, in collaboration with, of course, Dr. Debbie Knapp in College of Vet, and thanks to the drugs that are provided by the research, and the support for Purdue Institute for Cancer Research, who spearheaded this particular clinical trial, we are now running this clinical trial with dogs which have aggressive bladder cancer. So that's great. The second part that I would say, like, this was one particular case. Is that something that is repeated? And what stood out to us was another drug in the list called cycloprox. And this is a typical antifungal drug that is topical antifungal, and you get athletic good, and people apply for getting rid of that fungal. So to us, this was surprising that, oh, an antifungal has an anti-cancer activity as well. So that's wonderful, because many of the cancer patients, due to going to chemotherapy, they also get a lot of life-threatening fungal infection. So if this drug was about to work, it could have a dual benefit. You could get rid of the fungal infection, and it could also help some of the anti-cancer activity. Going what?
It's part of the arrangement. We only do this with very specialists.
I like that it's going. That's wonderful. So going back, we have now this, that we don't know whether or not it has an anti-cancer activity. So the first thing for us was like, let's try it out. We took some of the kidney cancer cells and treated the drug. And we saw like, yes, there is a dose response effect of this drug, and it seems that there is some anti-cancer activity for this antifungal drug as well. Okay, so that's good. Going back to our original question, does it work inside the body? So we did again the same animal model, and this was thanks to the funding that we got from AXI. So thank you very much for providing the funding to do this particular study. So we took some of those very aggressive tumor cells and again placed it under the skin and watched them grow in the presence or in the absence of this antifungal. And we were disappointed. So we saw that like this particular antifungal didn't seem to inhibit the tumor growth the way that we anticipated it would. So we went back, spent a year to see what causes this, why this happened. Like we have like some level of inhibition in vitro or in the cell cultures, but when we go in the body, this doesn't happen. And turns out that this particular drug, this particular antifungal, gets inactivated in the body where there's a lot of iron or metal ions. So iron, specifically in a dose-dependent manner, inactivates the activity of this particular antifungal. So rather than giving up, we decided like, let's team up with Dr. Flaherty from medicinal chemistry to modify this drug a little bit to see whether or not we could make it more resistant to this iron inactivation. And indeed, we found a version that is no longer gets inactivated as much because of the addition of the iron or addition of any other metal ion to this particular segment. Tested back, and we saw like, yes, now that we have removed that dependency to the iron inactivation, it works reasonably well. So if you think that it can have a better anti-cancer activity. So now going back to our original intention, which was maybe these antifungals that have anti-cancer activity would give us a dual benefit. If we got rid of its antifungal activity, did we make it better? Is it now a better antifungal or not? So to test that, we collaborated with Dr. Suttriggs and established a mouse model of oral yeast infection, or so-called thrush. And this is very common in cancer patients. You can see like the thrush happens inside generally the oral compartment, and it's like a white film or yeast infection that shows up in the mouse. So we said like, okay, if we now treat the mice with a mock, which is no treatment, or with an yeast, what happens is that those animals that get the yeast, they no longer could eat food very well, and they start losing their body weight. So you can see that those animals that got the candida or the yeast infection, they start losing body weight within the few days, within 4 or 5 days. And if we look at, my apologies to show some of the pictures of the tongues, but these are the tongues from the mice that got no infection, and you can see that the surface of the tongue is pretty clear. And if you get the tongue and try to grow the yeast from top of the tongue, nothing grows on the plate on the right. Those that are infected, there is a clear yeast infection and a white film on the top of the tongue. And if you look at the things that grow in the dish from the top of the tongue, is a lot of colonies that are formed. Okay, so what happens to our original antifungal that was topical antifungal? Well, we tried it, and turns out that it didn't work. So we have the same film formation on top of the tongue, and almost same number of bugs growing on the top of the tongue. But the one that we have modified, now if you look at it, if we apply that one to the mice, even just adding it to their food, what happens is that they don't lose as much as weight, and you can see their tongues are much more clear, and there are probably an order of magnitude fewer of those bugs that are growing on their tongue. So it seems that we made both antifungal and anti-cancer effect of this particular drug, cycloprox, better. And partly, we realized that within the saliva of the mouth, of our mouse, there is also a lot of irons that are potentially inactivating, and that's probably the mechanism that the original antifungal was not working. And when we made it more resistant to that iron inactivation, now we see a much better effect. So, what's next? And then this is almost my last study. So I mentioned that for many of these drugs, we know how these knobs are up or down. Which is true. We have spent like billions of taxpayer dollars to identify these. But the reality is that this is just for a fraction of the drugs where we know how these dials are gone up or down. And even for those fraction of the drugs for which we know, we only know this in 1 or 2 cell types. So we don't know in all different cells how these things are going to work. So what we are now doing is using all this limited knowledge that we have, and we are training these graphical neural networks and deep learning models where for any drug that we treat any cells with to tell us how these genes are going to be dialed up and down. So that potentially later on helps us to do much better drug repurposing as we go about. So with this, I have a lot of collaborators and a lot of students, a lot of undergraduates that are wonderful and contributing to this task. And I would like to thank our internal providers of funding, PICR and AXI, as well as the research company and all of the NIH funding that we have. Thank you.
If you have time for questions, please.
I'm apparently too fast.
Majid, for the antifungal, maybe I missed it, but what is the target and what is the impact? Does it induce apoptosis or does it just inhibit proliferation?
It's a great question. So when we looked at the downstream targets of this antifungal, it affects both NF-kappa B pathways and hypoxia. So through this 2 particular pathway, it enhances the MYC expression. So the antifungal decreases the MYC expression and the proliferation. Yes?
For most of these drugs, you saw the tumor growth decline, but then level off with some residual loss of tumor. This suggests that there must be another complementary gene that's driving the rest of the tumor growth. How do you identify that guy and how do you find the drug to treat it?
That's a great question. I think that what we could potentially do in those kind of scenarios is to go back and say, like, those resistant and residual cancers, now what type of knobs or what types of genes they dialed up or dialed down, and match a new drug to them. So that's a little bit of iterative approach, but I think that's where we could get to the details. Does that make sense?
Yeah, no, I mean, someone needs to do that first.
That makes sense.
So both of those, they came up with hits for specific types of cancer. Did you try them against other types of cancer, or is there any things they might be more broadly active?
And that's a great question. So I think for the CDK9 inhibitor, we tried it against liver cancer, and it was not as effective. We tried it against cervical cancer, which was the hit, and it was quite effective as well in that particular case. For the cycloprox, we have not yet tried it against other cancer types. But my assumption is that its activity is quite general, at least in the context of urothelial cancers. Yes?
How do you achieve specificity of your drug? That is go in correct tissue and does not have effect on the healthy tissue and does not affect actually the metabolism of healthy tissues?
So that's $1 million question, and I think that's where many of the people are working to make targeted deliveries of a lot of these drugs. But remember, what we started with was being somewhat blind of all of these, saying that let's take healthy tissue or cancer samples that survived better and cancer samples that did not survive as well, and then compare them, regardless of whether there is healthy tissues in there or so on. So this drug potentially provides some level of survival advantage for those survivors. So I think that in this particular case, the specificity is somewhat implicit in the implementation. But you're right. Ideally, you want to more target these toward the cancer cells.
Could I follow this question? Did you check these mice which were fed with inhibitor, silicate-9 inhibitor? Did they have the same lifespan that normal mice without cancer?
So I think that mouse models are generally artificial models, and the cancer that we put on them very quickly grow to the level that humanely we need to euthanize these animals. So we cannot let them go for more than certain time where they hit that limit. And if you saw that blue lines where the tumor grew, those were like the limits of the tumor that we could grow before humanely we could sacrifice these animals. Great question. Yeah.
Going back to your big picture question of why do different people respond differently to the same disease? You've shown us, I think I'll say it in an elegant approach, to look at a genetic basis for that difference. But of course, even identical twins can have differences in their response to disease. Are you looking at as well environmental influences on these differences and how can you adapt your approaches to enable such questions?
So that's a wonderful question. I think in our case, we try to match the survivors to less survivors as much as possible, right? So we sort of like, we have a group of people who succumb to cancer, unfortunately, but they survived 15 years with the cancer. And a group of patients that got the cancer and unfortunately died off of it very quickly. However, they came to clinic at the same stage. They were about the same age. They were same sex. And everything, they were both non-smoker, etc. Everything that we could have matched, we matched. Whether there are other confounders, like microbiota, like other things that could have been influential, you're right, we have not looked at those, or we couldn't look at those. What we saw is like their final outcome, which was the things that defined the cancer. Yes?
Can I just add this question? Would that actually be used as an additional kind of factor that will actually make your drug spell? For example, now you have a drug that is working at a very high level. If you would know the variation between the long survivors and the short survivors is music, could you add music to the drug and make it work even better? So what kind of approaches are you planning in your longer terms to kind of have like multiple environment and drug kind of combination approaches?
That's a fair point. I think that the crowd already gave us a few of those solutions. Phil mentioned that we could definitely go for those residual cancers. Natalia mentioned that we could go after specifically the cancer cells and targeting them. And then whether or not we could now include some of the additional factors, that's a great point. In this case, we thought that any of those additional factors, the consequence of that would have shown up on your tumor or on the tumor. So whether we could directly go back and measure them, that's a good question. Yes?
So we got it's a related question, but it really mentioned where the data came from and how good are those data? And have you noticed irregularities or poor data in your data sets that you need to purge to make your AMI better? And how do you get all the other tumor types and all the other drug types to feed in?
That's a wonderful question. So we started with these large atlases that have been created across many different cancer types through big consortium like Cancer Genome Atlas, where NIH funded billions of dollars to create this database. And through that, they have tried to keep the consistency of these data sets. That being said, many of the computational approach and many of these AI approach, one of the good things about them is that they are quite resilient to noise. So even if some of these samples are noisy, they could discern the patterns that are potentially strong enough for switching the things that we want to see. So I'm always appreciative of the efforts that all of you do to put your data in public databases. Even if you don't see the big picture of the data that you generated at this point, someone along the way with powerful AI and machine learning tool is going to get all of that data and potentially see much bigger patterns. And that's what thousands of researchers, billions of the public funding has gone to actually generate that and send them to sequencing read archive, as raw as of a data that is, or simple genomics data that's out there. I hope that I've answered some of your questions.
What could you do if you had pharma data sets that we don't get in the public data sets? What could you do with that?
That's a great point. Well, I think that would be a wonderful resource if you could go back now to add that to our training and specifically to look for rare disease types that we don't get a lot of those data coming to these public databases. So you're absolutely right. I think that there is an untapped potential there that we might be able to utilize. Yes?
Very inspiring presentation, but I have two questions. So we have kind of different. So the first question, people are mostly dying from metastatic forms. Like, things like the breast cancer metastasis into lungs, and this is why people die. So I don't see, because like we're just looking at the killing the cells, okay, more cells you kill, they are less chance to metastasize, like whether you looked at any of that process, because there is a lot of biophysics of metastasis process. But other question is also for every cancer there is a protocol. So how your drugs are going to go into their kind of physician office, clinic, and how they will compete and be evaluated versus existing protocols.
Wonderful question. So for the first question, I think that I would like to argue that we might start thinking about the cancer as not we are going to cure it, but as we are going to manage it. Meaning that we are going to have patients that could live with the cancer normal life. So many things that we have, for example, with patients that are living with HIV at this point. And so our approach was like, let's find those patients that are naturally living so long and see whether there are specific things about them that we could potentially go there. But I think that the same approach could apply to metastasis cancer. So if we, for example, look at those cells that become metastasized versus those that don't, are there any drugs that we could match up to differentiate them? We could look at that particular aspect. So the second question goes back to the core of this types of approach, which we call it as drug repurposing. So the drug repurposing is finding a novel use for already approved drugs where the safety is already known. So many of these drugs, we already know that are in clinics and they are used for different applications. So potentially these types of computational approaches that could repurpose the drug would have much shorter time going from the computer to clinic. Hope that answered the question. Yes.
Majid, this was really nice. I wanted to kind of go back to your discovery of the original antifungal drug, because that was kind of a serendipitous finding, right? That cancer patients have thrush, and so they were treating them with the antifungal, and you happened through your analysis to see that it also had anti-cancer properties. When you figured out this drug, you said it influences or stimulates NF-κB, so which is anti-apoptotic but also pro-inflammatory. Have you ever considered combining your antifungal with some pro-inflammatory drugs to see if that residual cancer completely goes away?
That's a great question. So I think in the mice, what we saw was that there was that residual cancer was like, basically the cancer did not grow, right? So that was the kind of stable disease as we categorize it. So certainly the drug does not have the killing property. It has the suppression of the growth property. But you're right, in the context of immune system, so if it could potentially reactivate the immune system, that would allow for that. Unfortunately, we did not use any of the models that break the immunocompetent setting, but we'll try. Yes?
You know, in your analysis of those genes that showed a dramatic difference between survivors and non-survivors, you know, one of these and you found an inhibitor of it that showed a great effect. Have you ever tried looking at a couple of the other genes that show these dramatic differences, adding them to the first to see if they complete the process of cure?
It's a great point. So I would like to mention that the CDK9 itself was not different in between those two patients, but what it was different, the things that would be dialed up or down by CDK9. And therefore we saw like, oh, now we can use CDK9. So that's why originally people would have not used CDK9 inhibitors. They're like, oh, CDK9 itself is not different. Why should we use this? So there's no logic to using it. Except we saw like those things that are the targets of CDK9, those are the ones that are changing. And then we went after the inhibitor. But going back to your talk, whether there are other to your question, whether there are other things that could be combined, we have started looking at the combinatorial effects of multiple drugs. And we have a great success of now at least finding the drugs that push the survival even further in a not additive way, but more synergistic way. So hopefully one of them would come up as a good combination. I hope that answered your question.
Yeah, I just wondered if they correlated with the genes you saw the greatest differential expression.
So not the gene itself, but the gene's activity. So meaning that like the downstream activity of the gene, there was a very good correlation. Right?
So, getting back to the question here about metastasis. So metastasis obviously will potentially do the dying work. Now your gene, now yourself that will be the growth phase of cancer versus the metastasis phase. Now at the slow growth phase, you can still have metastasis. Okay, metastasis doesn't necessarily depend on the rate of growth. You could be reducing the growth and the cells could still metastasize and go somewhere else. And their diet is different. You're expecting your CDK9 inhibitor to still work on that tumor? Because if it does not, then what you have done is you have reduced the tumor originally, but it's now growing a different type or something else. So how are you going to handle it?
That's an absolutely a fair question. So what I think is going to happen, like a lot of drugs of this sort, is that eventually patients are going to develop resistance. And that's when the metastasis would start occurring. So I don't think that this would be again a cure. This is a method to be able to enhance the survival as much as possible until the next step. I hope that answered your question.
Yeah, I mean, I'm just trying to see, like, there's a lot of expansion of the idea possibly, in the sense that if you can find the metastasized cell and find the dial of that, you could combine
And
the
we are
growth
looking at
phase
that.
and metastasis.
And we are looking at that for sure. Yes, Ben.
I can imagine that like an antibacterial, to be an ideal antifungal, you want to maximally impact fungal cells and minimally impact human, mammalian cells, right? So given that, were you surprised that an antifungal has this effect, considering I would imagine it's selected to minimally affect the cells that are your target, which are derived, cancer cells derived from mammalian cells, and does that make antifungals or antibacterial agents less of attractive candidates in the future for this kind of thing?
Good question. So I think that there are different classes of antibacterial and antifungals that are being developed. Some classes are specifically designed to target those knobs or genes within the pathogen that is just specific to that.
Right.
These classes of antifungals are not in that class where they are specifically targeting one gene. So the way that generally these works are through like azole pathway, but for this particular one is through chelation of the iron or chelation of some of the essential nutrients for that pathogen, which is much more reliant than that nutrition when it goes inside. Yes?
Maybe it's not very scientific question, but on your slide you showed very good drugs against pancreatic cancer.
That's right.
But why is that? This is the most difficult cancer to treat, and survival rate is very, very short.
Well, we have made, well, by we means scientists have made quite a lot of progress even on that. So the survival from 5-year survival from 5-7% has gone over the past probably less than a decade to 13-14%. So the progress is slow, but the progress every year has been made. That being said, that drug that you saw on the top of the list last year was approved as the first line of therapy against pancreatic cancer. We were actually very excited about trying that and then already approved, so we just dropped that one off the list.
And is it synthetic drug or it's naturally produced on plants?
Good question. So it is a topoisomerase inhibitor, and
Well, no further questions. It's been a very interactive conversation here. Let's thank the speaker again. Thank you.